So, the importance of proteoglycans is to extract water to form gels in order for it to keep the cells hydrated and cushion tissues against hydrostatic pressure. Proteoglycans can bind to a variety of ECM components, which includes growth factors, structural proteins, and cell surface receptors. Proteoglycans as well are cell type specific, and it is developmentally regulated.
The linkage of GAGs to the protein core in most but not all proteoglycans involves a specific tetrasaccharide linker composed of glucuronic acid residue, 2 galactose residues, and a xylose residue. Take note that this tetrasaccharide linker is most commonly seen in proteoglycans that contain heparins, heparan sulfates, dermatan sulfates, and chondroitin sulfates. Next, this tetrasaccharide linker is further coupled to the protein core through an O-glycosidic bond to a serine or threonine residue in the protein. Simply, we can say that a proteoglycan has a GAG, GlucA, GAL, GAL, SIL, SIR protein structure as shown in the figure. Glycosaminoglycans or GAGs forming the proteoglycans are the most abundant heteropolysaccharides in the body. They are long unbranched molecules containing a repeating disaccharide unit. Usually, one sugar is an uronic acid and the other is either a N-acetyl-glucosamine or GlucNAc and the other is N-acetyl-galactosamine or GalNAc. One or both sugars contain sulfate groups, and the only exception for this is the high alluronic acid. The protein cores of proteoglycans are rich in serine and threonine residues, which allows multiple sites of polymeric GAG attachment. Following the formation of the tetrasaccharide linker, if the next sugar added is N-acetylglucosamine or GlucNAc, the resulting attached GAGs will be either heparins or heparan sulfates. On the other hand, if the next sugar is N-acetylgalactosamine or GalNAc, then the attached GAGs will be either chondroitin sulfates or dermatan sulfates.
Let's talk about the heparan sulfate proteoglycans, or what we call as the HSPGs, which are present in the basement membranes where they collaborate with other matrix components in order to define the basement membrane structure and to provide a matrix for cell migration. The HSPGs are found in secretory vesicles, mostly in the glycine, which plays a role in packaging granular contents and maintaining proteases in an active state. It also regulates various biological activities after secretion such as coagulation, host defense, and food repair. The HSPGs can also bind to cytokines, chemokines, growth factors, and morphogens, protecting them against proteolysis. This also facilitates the formation of morphogenic regions essential for cell specification during the development and chemokine regions involved in the leukocyte recruitment. The HSPGs can act as a receptor for proteases and its inhibitors, which regulates their spatial distribution and activity. Lastly, the membrane of the HSPGs act as an endocytic receptors for clearance of bound ligands, which is especially relevant in the lipoprotein metabolism in the liver, and perhaps it also involved itself in the formation of the morphogenic regions during development.
Alright, so now let's proceed to the synthesis of proteoglycans, and we'll be using heparan sulfate as an example. It all starts with the translation of the core protein in the rough endoplasmic reticulum. This core protein will then serve as a heparinoid acceptor. This is followed by the synthesis of the N-link oligosaccharide acceptor. The core protein will then enter the Golgi apparatus, where the glucuronic acid, galactose-galactose xylose-serine linkage region is added to the serine glycine sites on the core protein. Next, there will be polymerization of the glucuronic acid or GlucA and N-acetylglucosamine or GlucNAc to form the polymeric form of the heparinoid. The basic sugar structure is modified by a series of sulfotransferases and carbohydrate-modifying enzymes, which results to complex sulfation patterns. And these sulfation patterns are critically important for the binding of heparan sulfate chains to proteins.